Our next speaker is Sung-Yon Kim from Seoul National University. He will be introduced via video by his former Picower mentor, Associate Professor Kwang-Hung Chung, and his talk is titled Warm and Full, the Neurobiology of Innate Behavior.
Good afternoon, everyone. My name is Kwang-Hung Chung, and I'm a core member of the Picower Institute. I'm delighted to introduce my dear friend and the first postdoc of my research group, Professor Sung-Yon Kim. Sung-Yon received his bachelor's degree in chemistry and biological sciences from Seoul National University in 2009. And then he moved to Stanford for his PhD training in neurosciences under the mentorship of Carl Dysers, where he studied neural pathways controlling distinct features of anxiety. Following his graduation in 2013, Sung-Yon joined my group in the Picower Institute for his postdoctoral training and developed many novel technologies for 3D molecular imaging and analysis of the brain. In 2015, Sung-Yon started his own lab at Seoul National University and has been studying the neural circuits underlying behavior regulation of homeostasis. He has won numerous awards, including Asan Award in Medicine, Hanseng Science Award, and has been named a member of Young Korean Academy of Science and Technology. Today, Sung-Yon will speak with us about the neurobiology of innate behavior. Without further ado, please join me in welcoming Sung-Yon.
All right. I'll just put it on the laser pointer here. Okay, so I'm deeply honored and excited to be a part of this special event for the 20th anniversary of Picower Institute. And obviously, I would like to thank KC for the opportunity and all organizers, including Liu Hei, and special thanks to Stephen for saving my life just before. My lab's current research interests are in the neurobiology of innate behavior, and more specifically, we are trying to answer the questions like, how do we turn on the heat when it's chilly, and how do we stop eating when we feel full? So these are the kind of things that we experience daily, and it's so mundane that the question sounds very simple and straightforward. But the truth is that we don't really know much about how these things are actually happening. So as a neurobiologist, I'm seeking scientific explanations for these innate motivated behaviors for the basic physiological needs. And just one caveat is that my talk is designed to be super lay friendly, maybe more on the extreme on this side. So we can talk more about the science, heavy talks afterwards. It might be pretty obvious for some of you where my motivations came from. I mean, my motivation for studying the basic physiological needs, such as warmth and fullness, it certainly has its origins in my time at MIT. So these are the photos from 2013 and 14 when I was a postdoc in the Chung lab on the 5th floor of the Picower Institute. My car was snow covered, but I still made it to the lab because I lived nearby and walked to the lab anyway. It's a fantastic setting for getting focused on the science and work. I felt very warm and cozy whenever I arrived in the building because the heating is so awesome in this building here. And on top of the warmth, I get this amazing free food from within the building. It's so warm and full. And just to be fair, this was one of those exceptional free foods, even with our high standards. This photograph was taken from September 9th, 2013, probably marking the Picower Institute's 11th anniversary. So here we are now. And in this warm environment, well-fed scientists make true discoveries, important discoveries, true innovations and inventions occur. So this, I believe, is quite representative of my time in the Chung lab, where KC sets an example for his trainee every day by stuffing the entire bench first, and then he miraculously makes the never-working experiment to work. And he probably displays it to us before disappearing in a rush of wind. So there I learned the bold, fearless kind of research style, setting the bar as high as possible and going to whatever lengths to attain it. he established so many huge and minor goals that they all seemed unachievable to me. We fought over so many like concepts and projects about the viability and things like that. But he actually demonstrated he makes things happen. He actually delivers things. So that immense bravery, the courage to come up with truly unique ideas, and the courage to raise huge questions. That's something that I really learned from KC and also from Picower Institute and MIT in general. So when I left MIT to build my own lab at Seoul National University, I took this lesson to heart, and I looked about to see where I could make the biggest contribution in science. And I noted that innate behaviors such as seeking warmth or eating appear to be something that we should have a complete understanding of by now in 2022, when we can send a rocket to space and bring it back intact, almost. So I began to concentrate on these issues, and so far I'm very happy with my own decision. These are the behaviors that are very well conserved across the animal species and can be readily studied in mouse models, and we can also be highly accurate and quantitative. So we can set the ambient temperature to wherever we want, and we can give them the exact amount of calories or volumes of food, and we can precisely measure the changes in body temperature, food intake patterns, when and how much, and what, and so on. So in the next few minutes, I'll go over the discoveries on these topics achieved from my own lab in the past several years, which were made possible by my experience at MIT, Picower, for which I'm eternally grateful. So first, seeking warmth, or now I'm going to say thermoregulatory behavior. So what you see here is a mouse repeatedly turning on a heat lamp by poking its nose into the hole or active port. And this is what we call a self-feeding task or self-feeding operant task. Each nose poking will turn on the heat lamp for 2 seconds, and the entire chamber is inside a refrigerator, so it's freezing cold. So this mouse is highly motivated to perform this operant task voluntarily. And as I already mentioned, thermoregulatory behavior is a basic motivated behavior for body temperature homeostasis. And this fundamental behavior is very well conserved across the animal kingdom. And obviously, this is because maintaining the body temperature within a certain narrow range is crucial for health and survival. In the face of a thermal challenge, the brain prompts an array of autonomic and behavioral responses to cope with thermal challenges. And these are some examples of autonomic responses to cold environment, which are highly stereotyped and involuntary. And I'm going to argue that these remarkable pioneers in the field have extensively studied the neural mechanisms underlying autonomic thermoregulation over the past few decades. And just to point out some important parts from the scheme, thermosensory information from the body first enters the brain at the parabrachial nucleus, or the PB, which then relays the thermosensory signals to the preoptic area, or the POA, a very well-established forebrain center for autonomic thermoregulation. And these neurons connect with certain hypothalamic area and other hypothalamic hindbrain areas and to the organs and tissues that actually mediate thermoregulatory responses. But in contrast to autonomic responses, behavior responses, or thermoregulatory behavior, is a motivated, goal-directed behavior that can be flexibly manifested in diverse forms, such as moving to a warm place, building nests, turning on a heater, drinking warm coffee, or wearing jackets, and so on. So these behavioral strategies to cope with thermal challenges are more energy efficient in terms of ATP, and thus represent a preferred mode of thermoregulation in animals. But despite its obvious fundamental significance, its underlying mechanisms remain strikingly unclear. And this is not just what I'm saying. This has been pointed out in several reviews. For example, this is a recent review by Zach Knight at UCSF. Thermoregulatory behavior remains the most enigmatic of the classic motivated behaviors, including eating and drinking. And no forebrain region or cell type has yet been discovered to be required for these responses. So jump to the conclusion. This is what we found. We found that a brain region called the lateral hypothalamus is generally required for diverse thermoregulatory behaviors. And furthermore, we found that a population of neurons marked by the expression of the VGAT gene is the one that's mediating this thermoregulatory responses. So these are some of the data. So we used a technique called chemogenetics to inhibit the activity of these neurons. And this manipulation impaired the self-feeding operant task that you saw earlier from my movie. But this task requires the mouse to be trained in task. So In other words, it requires learning. So how about the innate thermoregulatory behaviors? So this is one of the tests that does not require any training or prior learning. So in this thermal gradient test, a mouse is placed on a longitudinal chamber with a stainless steel floor with a thermal gradient from cold to hot here. So the mouse is free to sit on any position he feels comfortable with. And we found that chemogenetic inhibition of LH-Figure neurons also impaired the mouse's ability to select the optimal temperature for its body. And mice like to build nests when it's cold, and I like it too when it's cold on my bed. Nest building is another instinctive cold defense behavior in mice, and inhibiting LH-Figure neurons also suppressed this nest building behavior. And finally, postural extension in hot environments. It's literally just extending the posture to increase heat dissipation by increasing surface areas, and also decreases the heat production by muscle activity. And inhibiting LH-Figure neurons also inhibited postural extension. So I'm going to argue that these results demonstrate the identification of a forebrain region and also a cell type that is generally required for diverse thermoregulatory behaviors. We have so much more data that I can talk about for an hour, but just in the interest of time, let me show you just one more piece of data that I think is most interesting. So we observed these neurons' real-time activity using a microscopy technique called two-photon microscopy, which was already mentioned many times today. But we found that roughly a third of the LH-Figure subpopulation appear to inversely encode the thermal reward, thermal reward value of a thermal stimulus. That is, these neurons were strongly activated by thermal punishment and aversive thermal stimulus. That is a heat given already in a hot environment, like 100 degrees, like 37 Celsius. But the same neurons were inhibited by a rewarding thermal stimulus, like the same heat stimulus given in a cold environment, like 4 Celsius, something like that. And these neurons were largely separate from another group of neurons that happens to be also about a third of this population that are activated upon the consumption of the caloric rewards. So unfortunately, we are not able to find the specific molecular marker for these neurons, but we are trying to set up the two-photon, multi-photon stimulation setup that probably is similar with what the sort of lab might have now to manipulate these neurons' activity without having to rely on the genetic marker to determine their causal functions and others. So. Okay, so and then moving on to the next part. So in this study, we sought to answer the question, how do we stop eating when we are full? So like this guy eating a burger, when we have an appetite, we are driven to find food and find and consume food or drinks. And as we eat and drink, the ingested food and water exert mechanical forces against the digestive tract walls from the mouth to the pharynx, and then the esophagus, and then the stomach, and so on. So the mechanoreceptors in the digestive tract walls convert these mechanical stimuli into the neural signals. And these signals are transmitted to the brain to suppress appetite. It's like the gut saying to you that it's enough. You got to really stop eating. I'm full. So this basic physiological phenomenon has fascinated scientists since as early as 1800s, which led to numerous important findings over the past century. For example, this 1957 study experiment by Miller, it's another Miller, Neil Miller, just to clarify, used an intragastrically implanted balloon and found that gastric distension suppresses appetite. And also, this phenomenon has been clinically utilized in the recently approved gastric balloon therapy for treating obesity. And the inflated balloon inside the patient's stomach induces an early feeling of satiety during a meal and helps losing weight. So the phenomenon is there. We experience it daily. It's a fundamental physiological response, and at the same time, it's extremely important for health and survival. It can offer clues to treat the currently ongoing obesity pandemic in the world. It's already being clinically utilized already. But surprisingly little is known about the neural underpinnings of this phenomenon. So, and I know I don't have much time right now, so I'll just get to the point. So we found that a group of neurons in the parabrachial nucleus, defined by the expression of the gene named prodynorphin, P9 gene, monitor eating and drinking using mechanosensory signals arising from the upper digestive tract. And upon the receipt of the mechanosensory signals, these neurons produce appetite-suppressing signals that serve as a negative feedback on ingestion to prevent harmful overconsumption. Okay. To show you some data, obviously, all these experiments are done in mice, and PBP9 neurons are acutely activated upon water intake. And we also observed that they are not only activated by just the consumption of the water, but also a variety of other kind of liquids as solids, like child food or high-fat food or hypertonic saline, even silicone oil and something like that. And these neurons were also activated when we directly touched the mouth, the oral cavity and the pharyngeal cavities. And were also activated when we inserted the gavage needle through the esophagus to stretch the esophageal walls. And also activated when we inflated the balloon and implanted inside the stomach in a classic study style. And our two-photon experiments of these neurons activity revealed that the same neurons are that was activated by oral probing is also activated when we inflate the gastric balloon. And the same neurons are also activated when it's naturally eating or drinking and stuff like that. So these results really support the idea that these neurons can monitor, we don't know it does, but it can monitor the ingestion using mechanosensory signals arising from the digestive tract. Okay, and we further showed that chemogenetic stimulation of these neurons potently suppresses food intake in hungry mice and also suppresses water intake in thirsty mice. And chemogenetic inhibition of these neurons induces pathological overconsumption phenotype. So this is the mouse version of the summary describing a neural circuit mechanism for mechanosensory feedback control of ingestion centered around the PB prodynorphin neurons. And because I'm from the Chung lab, obviously, of course, we did the whole brain clearing and imaging. And this is a hemisphere of the mouse brain that expresses green fluorescent molecules in the PBP9 neurons. And you can see sort of, bifurcating, pathways, the one that goes up to the perioctal gray, and the other pathway going down through the midbrain areas to pretty much all the subregions of the hypothalamus as well as the preoptic area, somebody calls it preoptic hypothalamus, and make a sharp U-turn to go to BNST and other brain areas. So we are currently exploring the function of each downstream pathways, especially with the focus on identifying the region that integrates the hunger signals with the feedback fullness signals. Okay, so with that, I'd like to thank my students who have done all the work that I talked to you about today. And I would like to also thank my lab members, colleagues, and funding sources. And in the spirit of the occasion, I'd like to also thank KC and the Picower Institute once more for all the opportunities that I've had. And it was a true privilege to be a part of the MIT community and also be back in here and given the opportunity to speak about our work. So thank you so much for your attention. Sorry about the technical difficulties. Okay.
Thank you, Sung-Yon. Questions? You're putting me on the spot again. I'll ask. Sorry, question, please, go ahead.
Yeah. So I noticed as you were speaking that it seems from my memory that there's a ton of overlap between these kind of circuits that you were discussing, like for feeding and satiety, with the ascending arousal system. And I was wondering if you could kind of talk about how those various kind of regulatory systems interrelate with one another.
Right, yeah, so that's a good question. And so we have been studying several brain areas, including the PB prodynorphin neurons. And we focus just on the function of the PBP9 neurons in mechanosensory feedback control of ingestion. But around this very similar time we published, our paper went out. Several other groups published the paper on the PBP9 neurons implication in pain signaling and also some other signalings like arousal and thermoregulation and stuff like that. So yeah, I think it's a really good question that we should all be asking after, because most of the papers, including myself, just look at one function at a time. But even for the lateral hypothalamus brigade neurons that we have been recording for the thermal punishment and reward and calorie rewards, there's obvious heterogeneity. And so I've been adding more and more stimulus and see what exactly the subpopulations of neurons encode. But then, you add more, sort of the behavioral modalities or functional modalities into the paper, into the experiment, and the data becomes much more challenging to interpret. So I've been talking to this a lot to the folks that I started to meet in person on the conferences this summer. And yeah, most people acknowledges this, that when you look at just one function, it's pretty story. But if you look at try to account multiple functions, it becomes more difficult. But yeah, I think we should really be doing that too. But yeah, but regarding the arousal itself, I think if you stimulate the pituitary neurons, then it's going to increase arousal rate, arousal level. And also there are a lot of overlaps between arousal and feeding. Like LH3 neurons, obviously it's going to drive arousal, increase arousal level. And even like LCNA neurons, those neurons are activated upon feeding. So what are these neurons doing? Are the same individual neurons wearing multiple hats, or are there subpopulations? Those are I think that's the sort of the question that all the community is seeking after these days. Thanks.
Sung-Yon, thank you again.
Yeah,
Thank you very much.
Thank you. Yeah.